Wednesday, you need to make sure you bring this. Today, I hope you brought it anyway. But on Wednesday, you'll need this for your diff. Does everybody have their cards made out? Does everybody have their cards for WBC estimate, platelet estimate, morphology with the indices? Do you have those? If you do not have those cards, please don't come to class, because you're just gonna take way too long. Okay, so can you get that done before Wednesday? Any questions about that? So the slide I'm gonna be giving you is a known slide. It has a cover slip on it. So you'll focus on 10 and then immediately go to 40 power and do an estimate. I will be skirting around the room to each one of you and constantly showing you, when you raise your hand, where to go. So think about that lab and like where we had to go on the slide and where we don't want to go on the slide, and that will really help you. Okie dokie. Alright, so the next thing is I kind of want to go from the beginning of what we're going to be talking about today. But let's do just a really quick recap. We have our pluripotential cell. This is the multi-potential or pluripotential cell. Everything is coming from this cell. It's going to go into two routes. What are the two routes that are the progenitors? Okay, so myeloid, and you don't need to do this in any certain left or right. You could do myeloid over here, it doesn't matter. Or lymphoid. Does anybody want to write this down again, or you got it? From the lymphoids are going to make T and B lymphs. T and B lymphs. Now, both of these are very important to our immune system. This requires prior recognition, meaning it's an adaptive response, meaning it comes from something that it's seen before, and it's going to make copies and try to kill it, or going to create what? What does the bees do?
Antibodies?
They make antibodies, right? So bees really turn into mostly, remember this, plasma cells. And from plasma cells we know to get antibodies. Everybody good on that? Basic immune stuff, right? Then we have our myeloid. And from here we have a little thing called the what?
Gem.
The G, the E, the M, and the M. So one of these M's is going to be the megakaryocyte. The mega meaning really big, karyocyte. Now it really is a wild thing because it doesn't progress like we know and love in the way that cells progress, start out big, get smaller, lose the nucleus, that kind of thing. It's a very strange way that it makes platelets, and we'll talk about that in a couple weeks, but these are just simply platelets. The other M is a monocyte, and so today we're going to be talking about the granulocytic and the monocytic series. What's on the board? These are what? These are all segmented neutrophils. This is 70-80% of what we have in the diff, right? 70-80% of these is what we should have. They all have sausage in them. Does everybody see that? It looks like linked sausage. Those are held together, these sausages are held together by filaments. Okay, but we're not there yet. No mono pictures up there. Monocyte, and then this is a erythrocyte, and this, you could just say an RBC or an erythrocyte. We've already talked about that progression, haven't we? Then the G. Now, there are 3 types of granulocytes. They all, and listen up, they all look like this. They all have sausage, but they have different colors. They all look like sausage. So the granulocytes all look like the same. They all look, the nucleus looks like sausage. Okay, I equate everything to food. I like to eat. Granulocytes. So the three granulocytes are the segmented neutrophils, which are those, 70 to 80%. The what? Eos, which are, you can write down the amounts. How much Eos? Anyone? Oh, come on, guys. This was a couple weeks ago. About 1 to 6%, and then finally, basophils. You guys are going to be seeing this on Wednesday, right? So you need to know your amounts. You need to know your amounts. Basophils are 0 to 1. These Eos are colored what? What kind of granules? Red, orange. Basos are blue, purple. So they look like this, except these are segs. They look like this, except what? Eos have orange, and basos have, can you go to your book? Can you go to page 9? Please go to page 9. If the lights aren't on high enough, we'll turn them on higher. This is the seg on the very first row on page 9. Actually, you should have your books already out when we're gonna start lecture. It's always helpful. Okay, so page 9. Does everybody see the seg in the first row? The immature seg is the band, that's in the second row. Now skip down. Does everybody see the EO? That's in the 5th row across. Does everybody see the orange? But do you see the sausage? And then how about that baso? It's hard to see the sausage with the purpley blue granules, isn't it? It is hard to see, I admit. But those are actual sausages in there. Not actual sausages, but they look like sausages, right? Okay. The monocytes is on the 4th row. Those don't have any granules. There's a really pretty one in the Turgeon text of monocyte on page 173 and 172 how the mono matures. Okay, so 169 has your maturity, 166 has your maturity. So let's stay on 166 on Turgeon, please. Okay, so how many monocytes in the peripheral blood? If I count 100 cells, how many monos would there be? Because we already did segs, eos, basos. How many monos? Somewhere around 3 to 11%. And then how many lymphs? Not T's and B's. We do not classify it as a T or B. We just call it lymphs. How many lymphs do we have? Yeah, 20 to 40. So somewhere around 30% or so. 20%. Okay, kids have higher lymphs. Why do you think kids have higher lymphs? It's an adaptive response. It's survival of the fittest. Why do kids have higher lymphs? Lymphs fight what?
Infection.
Not bacteria. Viruses. Kids are exposed to viruses all day long, right? So lymphs are higher percentages in children. So if you look at the back of the book and you look at the appendices in the back of the book, you'll see that kids and adults have different types of values, right? For a lot of reasons. They have more red cells because they need more oxygen because their tissue's developing. Their white counts are generally higher. Why? Because they're fighting infection. They need to fight infection. It's an adaptive response, right? It's survival of the fittest. These little ones need to survive. Okay, so are we good with that? All right, so that's a quick recap. So again, these are things that you should know. You need to write them down on a card, please. Please have your cards ready for Wednesday for your lab. All right? So now let's look at the progenitor cell. What we're talking here is the myeloid and then specifically the G. Is everybody seeing where I'm at? We're at myeloid and now we're specifically at the G. And so we have these colony-forming unit granulocyte. And you're like, how do these things know to do what they do? Well, there's a lot of cytokines or chemicals or proteins. They're all the same. They are programmed within us to tell these cells what to do, okay? So we can talk and list all the cytokines and these proteins out and hash these out all day, but I don't know them all and you're not going to know them all. It's not important. What is important is that you know that they're coming from myeloid. We're talking about granulocytes specifically. And now we're talking about the seg specifically. This is the seg. Specifically what's going to happen is we notice that we got the myeloid metaband segmented. Yes, it could be neutrophils, eos, and basos, but I will tell you this right now. So it's very, very rare. It's extremely rare, and you might want to put this in your notes. This whole thing would be so rare to see an EO or a baso immature in your cells, like on a smear, when I'm looking at the diff. It would be extremely rare to have an EOs that are immature or basos that are immature. Our leukemias that we have are not usually basophilic or eosinophilic in nature. So this is why I'm saying hone in on the fact that it's probably just a seg, meaning 70 to 80% of the cells that we have in our diff are segmented neutrophils. So the leukemias that we see is one reason why we would have these immaturities. Everybody knows what leukemia is. It's a white cell cancer. Now there's different kinds of leukemias. You can even say there are platelet cancers and there's erythrocytic cancers. Yeah, there is. But when I say leukemias, we usually say it's either myeloid in nature or lymphoid in nature, a person that has leukemia. Okay? But to have an eosinophilic leukemia or a basophilic leukemia is rare. So where will we see these immatures? We will see them when the segs are young. These segs are young. The 70 to 80% are young. Is everybody understanding what I'm saying? Cool? Any questions? If you have questions, raise your hand. Don't sit back. I will repeat it. Okay? So, we as technicians on a slide, when we're doing the diff, we're expecting to see what? We're expecting to see 70, 80% of these segs, but in certain situations like infections, because these are bacteria, granulocytes eat. This isn't virus. This is eating. This is bacteria. So what are we seeing? We're seeing this immaturity. So why? Most of the time, again, infection or some sort of cancerous state. Is everybody cool on that? So we do need to know the names of these. So do you remember PBPO, peanut butter pope? We do need to know the names of these because you see these every day. In the lab, in hematology, you'll have to identify these probably every day. And when they get immature like this, and when you start to see the immaturity, then you have to refer to the pathologist. You can't report the diff out when you have this immaturity. You have to send it forward to the pathologist so he or she can look at it and verify it. Does that make sense? because they're immature. It's pretty bad if you have that, right? So you can say, oh, well, I've got 5 myelocytes. Any immaturity at all like that's going to go to the path. Okay? So, is everybody good here?
You have to say to memorize that one.
You have to. This is a must. You have to memorize these cells. So, anything early is blast, right? Myeloblast. Pro-myelocyte, meaning coming before the myelocyte. And then meta is changing. Meta is the kidney bean. That's changing. Now I'm at a kidney bean. Band is a C or an S shape. A C or an S shape. And the segmented is segmented. It's truly segmented. It is normal to have bands 1 to 5%. It is normal to have bands at 1 to 5%. So do you want to write that down maybe? It is normal to have bands that look like a C or an S at 1 to 5%. And you're like, well, what's a band? Well, let's look. It's so normal to have bands at a 1 to 5%. Look on the second row. Does everybody see the second row? That's your band. It's okay to have 1 to 5% of these, but guess what, guys? Look at the difference. It has to be a C or an S shape. If there's any overlap, so the rule is this, if there's any overlap of segments, so when the cell gets smeared out, because you all made smears, right? You made smears. You made smears. When the cell gets smeared out, if the lobes go on top of each other and you can't distinguish, guess what you do? You give it the benefit of the doubt and you call it a seg. The only time I can call it a band, the only time I can call it a band, one of these in this row, is if it is a true C or an S shape. So please write that down. The band can only be a C or an S shape. If there's any kind of overlap or any kind of guess, like, oh, it looks like it's pinching here. It looks like it's pinching and making a little filament. Can't call it a seg. I mean, you can't call it a band. Sorry. Yeah.
What about the last one, the second row? That's kind of like
That's tricky, but there's really no I don't see any overlapping sausage. There's no filament at all. So see, the thing is if it does that, you have to say, are any of those lobes overlapped? Those 3 lobes are not overlapped, but there is no filament. So that's a good question, Alex. Does everybody see the one that Alex is talking about? So it depends on the cell at the time. Remember, you're on 100 oil. You're on 100 oil for a reason. It depends on that one like how close I could see. If I could see in the nucleus and make sure there's no overlap, Alex, then I would make sure I would call it a band. She's calling it a band here just to give you an idea that there are 3 really lobes. It doesn't look like the lobes are overlapped. It doesn't look like there's a filament. The filament is the thing that joins the segments together, the little thin thing. So very good question. Let's start with the myeloblast. So we can look at Bunny at the same time, please. So what is this going to be on, guys? This is going to be on neutrophil maturation. So we start on page 41. We're going to be on page 43. So 43 is the myeloblast. Okay. So myeloblasts, everybody would definitely agree that that is a big, ugly cell. Yes. And it also has what inside of it? It does have some granules, but it has that nucleoli, right? The nucleoli, those round little structures. right? Why is that? What did I say? What's going on? What's going on? What's it doing at this stage of the game? Why does it have?
Growing.
Big brain.
It's got a big brain. It's growing. It's thinking. It's maturing. It's young. Have you ever seen babies with big heads? They got a big brain. I think those babies are cute. Courtney's like, really? Do you do? All babies are cute. But some babies have very big heads, don't they? So some of these myeloblasts are very big and ugly. No, babies aren't ugly, but never mind. Babies are sweet, but they are parasites. Anyway, everybody got the gist of the nucleoli in there? Okay. Promyelocyte. Now, what's going on here? The cell is, you can turn the page. And we also have our lovely book. So, at the same time, you can be looking at the table, and you can be looking at the maturation sequence over here on the left. See, this is the stuff that I want you to look at before you get to me. Because when you get to me now, you've seen it before. And you're like, oh, she's talking about this again. I just looked at this before. That kind of thing. Because these words are very strange and you've not heard of them before. So if you don't prep before you get to me, it's going to be lost in the sauce. Okay, so this is interesting. Look at the blue. Look at the cytoplasm. It's very, very striking. It also has these little purplish, look, these littleish purplish reddish granules. Those are called our azurophilic. Azurophilic. Liking, philia. Philia means liking. Azurophilic granules. The nucleus is doing what? It's a chromatin condensation, and the nucleoli are getting less prominent. The cell is actually a little larger than a blast, even though it doesn't look any larger, but sometimes it is a little larger. It's still big, it's still growing up, it's still trying to think. But does everybody see the nucleoli? Is it there? I can't see it from far away. Yes? Okay, good. All right, so that is your pro. Now, look at the difference. What happened to the cytoplasm, guys? This is the big one, not the smaller one. That's a lymph down there. The big one. What happened to the cytoplasm? Tell me something about it. There is more cytoplasm. Tell me about the color.
Much lighter.
It's much lighter. Tell me about the nucleus.
I guess smaller.
It's definitely smaller, and then it's doing what? Do you see the word eccentric? Very important word. Eccentric means, look at the nucleus. See where it is? Is it in the center?
No.
Eccentric means it's off center. Eccentric, it's off center. Isn't it pushed to the side? Eccentric, important term in our morphology. Eccentric means that it's pushed to the side. Again, the table is showing you the difference in the NC ratio and different things like that. It's actually showing you on page 168 different types. It's got a pro, a myelocyte, and a myeloblast on figure 8-7. Look at this at the very bottom, guys. It's showing you a slide that has a pro, a myelo, and a myeloblast all in one. So this is the kind of stuff that you would see in the lab. And then you would refer it. You would do your best. You would do your best on trying to identify. And you're like, oh my God, are we going to do that at Cincinnati State? What did I say last week?
Yes.
Probably not. Not a lot. Why? Because you just need to identify what segs and bands are. honestly. You might see a little bit of this in May and June. May and June, we might throw a couple your way, but we can't do that in Heme 1. Heme 2 is a dry lab. We're gonna look at slides, but we're not gonna actually do any microscope stuff in the spring. Okay, we'll look at pictures like this. And then in May and June, we might throw a few of these your way, but we just need you to do regular diffs and be really good at regular diffs, because in the summer, at late June, July, and August, you'll get these. They'll give you the diff box. They have this box in your lab. It's the famed diff box, or the abnormal box. And after they say that you're good at regular diffs, they'll start to give you these, and you won't be challenged. They have cover slips on them just like mine. You'll be really challenged when you're looking at these. But you can have your book out, you can have your atlas out, right, when you're at the clinical site doing these. So don't get your panties in a bunch yet. I'm telling you that it's okay. It's good to know all the normals before you start to see abnormals, because you can't call anything normal or abnormal if you don't know what's normal, right? The meta is the kidney bean. So if you look, wait a minute, did we go to myelocyte yet? We did? Okay. Now, wait a minute, did I make sure? Hold on a minute. Okay, so just make sure that you make a note for the myelocyte. On the note for the myelocyte, make sure that you say at this point is when the secondary granules can be recognized. So there's primary granules and there's secondary granules. The secondary granules are going to help us with when we get to the mature segmented neutrophil. The secondary granules can be seen, and so now they're starting to appear here. So at this stage, you need to make sure that you say, okay, the secondary granules are coming in at this point on the myelocyte. And so if you look at Bunny's book, she outlines the cytoplasm, the granules, the nucleus for you on here. Now let's go to the kidney bean. I like this one. You will see a lot of these because remember, this is the stage before what? So look at this page. What is this right before?
Band.
The band. Does everybody see this sequence? What did I say? It's normal to see how many bands? 1 to 5%. So wouldn't it be likely then, if you were going to see any immaturity, that you'd probably see some of these then? You have segs. You might have some bands. Now why would I have the next ones? Well, because it's immature. So this would be likely to see more metas than anything else after the bands. Should we see metas ever? What's the normal in 100? Should we have any of these? No, none. We have segs and we have bands, right? It's normal to have 1 to 5% bands. If you have a slide that has no bands, great, good, that's normal. You're like, well, you said it could be 1 to 5%. Yeah, but it's okay to have none. You don't have to have bands when you're looking. Can you have these? Yeah, if you have some immaturity, but is it normal to have any metas? No, it is not normal to have metamyelocytes. So if you had any of these, path review. Path review. Same thing with the bands. 1 to 5% bands. If you have more than 5% bands, most of the time they're going to say path review it, because it's not normal to have immaturity. Does that make sense to everybody? All right, so it's a kidney bean. It's a kidney shape. Now look at the cytoplasm now. What's it doing? It went from striking blue with those primary granules of azophilia. And now what's going on? It's getting more
Faint.
It's kind of faint pink now, right? It's almost taking like a little bit of a bluish tinge. Isn't it strange? The cell's actually smaller. Look at it compared to the red cells. The cell itself is smaller. The nuclear chromatin is tighter. Okay? So numerous secondary granules. So neutros, EOs, or basos clearly outnumber the primary granules. Okay, so the primaries are going away, the secondaries are coming on. The band. Now this is a platelet up there by the band. Does everybody see the platelet, the little purple dot? That's a platelet. This is a band. It's a C or an S shape. So you say to yourself, is there any overlap? I don't see any. That's a really good band, isn't it? But some of the ones would be like the one at the end that Alex was just pointing out. It's a little tricky, so you have to look. So it's a U-shaped or C or S shaped, and the chromatin is heavily clumped, and then the specific or secondary granules are there. Okay. Segmented neutrophils. By the way, this guy, does anybody want to take a shot at that? It's not a white cell. We learned about the calculation last week.
A nucleated red blood cell?
It's an NRBC. It's a nucleated red cell. Remember the nucleated red cell? Should we have any nukes in our bloodstream?
No.
No. When we have more than 5, we have to do what?
Correct the white blood cell
Correct the
count.
white count, because the cell counter, the instrument, picked these up and counted them as white cells, right? So then we have to do that calculation. This is a nuke. So while I'm doing the diff, I get another counter, like a silver counter, and I count these. And if I have more than 5, then I'm gonna have to make an adjustment on that white count, right? Okay, so this is a segmented neutrophil. Very many segments on here, but it has the filaments. So this is a good picture because it's showing you the filaments, or the threads that are holding the actual lobes together. Does everybody see the threads that are holding the lobes together? The cytoplasm is pink due to the large number of the secondary granules that we see. Alright, so now let's go into the monocyte maturation on page 55, but really page 56 and 57. So 56 and 57. In Bunny, I'm on 56 and 57. In Turgeon, page 169. No, sorry, my bad. That's the EO and the baso. It starts on page 169, the monocytic macrophage series. Page 169 at the bottom, the monocytic macrophage series, saying it depends on what macrophages are in the body. So like if it's in the bone, it's called an osteoclast. If it's in the liver, it's called a Kupffer cell. Have you guys heard of those before? Maybe in biology or anatomy, you might have heard of those cells. But what do macrophages do? They engulf, they eat, right? So again, we're engulfing and we're eating. Okay, now turn the page on page 170, and it's talking about monocytes in general. Okay, so the name changes with the location, and so again, if it's in the liver, it can be called a macrophage, right? It can be called a macrophage, but it's called a Kupffer cell. If it's in the brain, I think it's called a microglial cell. Yeah? trying to recall. Sound familiar? Yeah. So if you look at this, you can see the vacuoles in it. Can you see the vacuoles in it? The holes? Those are vacuoles. Vacuoles is a sign of eating. Vacuoles are holes as a sign of eating. And so we have these monocytic macrophage stages. Okay, so let's go back real quick. So let's look at the monoblast. So the monoblast, again, we should never really have these, but it is the earliest form of the monocyte. And then flip the page over. Then we have our pro-monocyte, and it's starting to really look like a mono because it's got that blue cytoplasm, but remember it's a gray-blue cytoplasm. And it has the nucleolus in there, in the nucleus. And then finally the monocyte. So let's look at the monocyte really quick on this page. On this page, what is it? It is page 61. So there's a couple things about monocytes. Does everybody see the vacuoles? A lot of times in a monocyte you will see vacuoles, and now you're like, well, wait a minute, it kind of looks like a lymph. I will tell you, on Wednesday, you're going to be like asking yourself, is it a mono or is it a lymph? Now remember, I'm going to say, on my board here I had, what? Monos were in with the myeloid, and lymphs were their own thing. Do they look a little similar? They can, but their function is totally different. So if you go, right, because monocytes are more bacterial in nature and other things, and lymphocytes are more viral. So if you look at your page, is it 9? Yeah, look at page 9, everybody. See the difference between your lymph and your mono? There's a real big difference on page 9 between with your monos. So let's look at all these monos. Let's look at the 5 monos she gives you. Wouldn't you agree that if you're looking on page 9 right now, the third row is the lymphs. The 4th row is the monos. On page 9, the cell is what? Actually, the cell itself is actually bigger, yes, than the lymph. How about the cytoplasm?
There's more in the
There's
lymph.
more cytoplasm. How about the color? The color is lighter. So the monocyte has a blue-gray. So monocytes have blue-gray, whereas lymphs are more blue. That is one huge distinguishing thing. How about the nucleus shape? The nucleus shape is way different, guys. The lymphs, they are what? Round to oval. And then the monocytes, are monocytes round? No, they're kind of like convoluted, aren't they? They're kind of brain-like. They're kind of kidney beanish, brain-like. How about the chromatin? The chromatin is not as thick, is it? The monocyte has more loose chromatin. Loose chromatin. So these are the things that when you get to the lymph and the mono, you have to ask yourself those questions. This is how pathologists would teach you. But except they're going to give you 5 minutes to learn it. And I'm going to give you the rest of the semester to learn it. Honestly, they'll say, well, why don't you know this? I have PTSD. Why do you not know this already? I don't know. I was not born knowing these things, right? But the key is to study the differences in the nucleus, the cytoplasm, the size of the cell, the color of the cell. Any good hematologist will show you and tell you that. So I'm telling you right now, those are the things. This is how we classify these cells. We look at the shape, the size, the color, the nucleus, the cytoplasm. Are there vacuoles or not? Now, lymphs can have vacuoles. It's not likely, but they can. Look at this lymph on the very end. It kind of looks like a mono, doesn't it? But it still is different for size, shape, color, cytoplasm, nucleus, but it has red azure-philic granules in it. Look at that lymph. So it's exciting, for sure, I think. I think it's very interesting to get to know these things. If you look at page 13, this is giving you that big picture. On page 13, isn't that neat? Now you guys almost know all these on page 13. What happens to a monocyte when it grows up? Because it only is out in the periphery for less than 24 hours. The monocyte is only out there for less than 24 hours. Where does it go? It goes to the different tissues as a macrophage. So when the mono grows up, it becomes a macrophage. So maybe you could write that in your notes, that monos don't live very long. They live like half a day. Monos live like half a day, and then they go out from the periphery and they reside in the tissues. They reside in the organs as a natural immune response to help eat, to help protect us, to help our brain, to help our liver, to help all the other parts of the body. So macrophages are out there fighting for us, so monocytes become macrophages when they grow up. Okay? Cool beans? Now, chapter 18. She switches from 8, and then we're going to go to 18 in turgid. So please go to 18 in turgid, all the way out to 18, please, chapter 18. So 18 is entitled Non-Malignant Leukocyte Disorders. What does this mean, non-malignant? Anybody? What time is it? What's non-malignant mean? Peyton? Not bad, not
Cancerous?
cancerous. So can you remember that right now, that this is not cancer? We're saving cancer till Heme 2. We'll discuss malignant white cells in Heme 2. So we're talking about disorders of these white cells now that are not cancerous. So 18.1 for leukocytic increases and decreases and examples. I'm saying know it. So if you look at table 18.1, everybody look at that on page 337. Page 337. Do you have your books? If you have your table there, the table is showing you increases and decreases in what we've been talking about, which are segs, yes, segs, eos, basos, and monos. So if I have the word philia, what does philia mean again? Liking, loving, a lot of it. A lot of it, yep. How about osis, monocytosis? Look down in the middle, monocytosis. A little or a lot. Monocytosis means a lot. So increase in monos, increase in basos, increase in eos, increase in segs. Now, let's look down from there. Neutropenia means what? Penia means decreased. So, segs that are decreased. So, how many segs do we normally have? 70 to 80%. So, what would be a neutropenia? Maybe if a person had 40% neutrophils. You can write right in your book or the table, but I'm kind of outlining these words for you. So again, neutropenia would be like, okay, you have 40% neutrophils instead of 70 to 80%. See what I mean? So I'm giving you an example. Eosinopenia. Well, we only have 1 to 6%. So maybe you only have 1 to 2% or none, right? Basopenia. Well, it's kind of good to have basopenia. But you're like, okay, well, you look at 100 cells and basophils are only 0 to 1%. You get what I'm saying? What they're talking about is not necessarily looking at the diff all the time. They're just talking about in the periphery across the board. If I looked at your smears and looked at your smears, and I never ever seen any basophils, right? Monocytopenia, no known conditions. Let's go to the very first one again, neutrophilia. This is what you will see the most of in the lab. So in your notes, you could say, I see the most of this in the lab, neutrophilia. That would be what? Beyond 70 and 80%. So an example of neutrophilia would be like 85%. Maybe even 82%, 85%, 90%, 95% neutrophils or segs. Lots and lots of reasons, right? Inflammation, infection, surgery, burns, stress, drugs, hormones, types of leukemia, absolutely. So eosinophilia, this is the second one you will see the most of. Now, isn't that neat? They did the table and how you would see that. So eosinophilia is allergies. So you will get this question probably on your registry exam. You'll certainly be asked by me about these conditions. But definitely allergies. If you have a lot of eos in your bloodstream, you probably have allergies or hay fever or something. Hopefully you don't have parasites, because parasites are another reason to have a high EO count. Does everybody see dermatosis? What does dermatosis mean? Skin condition. Okay? So, basophilia, like lots of basos, colitis, smallpox, sinusitis. Does everybody see the word leukemia? I will tell you this. A pathologist is extremely concerned when he or she sees more than 0 to 1% basos. So to remember, for basos, we should only have 0 to 1%. If you have 2 or 3 basos in your slide, when you're doing your slide, you must review it for path. You must review it for 2 to 3 basos, because the pathologist is concerned that you have cancer. Okay? So, we talk about, like, what are you normally? My husband, he runs a real high EO count, because he is allergic to dust, and he's constantly sneezing. I don't care what time of year it is. The man wakes up at 5 in the morning and just sneezes 10 times. So, he has a lot of EOs. So what do you have in your bloodstream? I don't know. It would be interesting to find out, right? Some people just run high monos all the time. We were talking about that today, last week, me and Alexandria. What are your normals? Maybe you run more lymphs than like 20%. Maybe you run always like 30, 35% lymphs. Maybe your mono, you're not running 3 or 4 monos. Maybe you're running around 10% monos all the time. You would just have to do your blood every 6 months or every year and look at it from year to year and see what your counts do. While you're normal, not while you're sick, right? While you're sick is not an opportunity to do a normal. So I'm asking you to please know these conditions. Now, I need to finish up here with some of the inclusions. Now, Bunny has these in her book, so I'm asking you to also look at these inclusions and your summary table 18.2. So vacuoles, vacuoles, and Dolly bodies. These are seen in infection. So, anybody want to help me with this on the Bunny? Let's look at vacuoles and Dollies so we can let everybody know. Shirari Kagashi.
It's page 134.
134? Okay, thank you. Okay, great. So vacuolation and Dolly bodies, page 134 and page 135, you can write that down, please. Page 134 and page 135. Why do we see these? Where are these seen mainly? In what? One word. In infection. Do you have to have both? No. Can you have one or the other? Yeah. Does everybody see the holes? Those are vacuoles. I spell it vacuoles. Okay? Dolly bodies have an umlaut over the, is it the O? The umlaut, the double dot?
Yes.
Is that an umlaut?
Yes.
Okay, whatever. I'm not an English major. Does everybody see the blue line? That is not a stain as far as like stain remnant. It's actually a Dolly body. It's a sign of infection. Now, it can be associated with other things like cancers and chemo and things like that. But both of these remind me of infection. Now, there's another one thing, infection. So write this down. Infection, I want you to think Dolly. I want you to think vacuoles. And I also want you to think toxic granulation. I'm not showing you that yet. Now, please write this down. Infection. So I'm thinking more bacterial, right? Dollies, vacuoles, and toxic grams. These are in segs. These are in segs. These are in segmented neutrophils. Now, when you have an infection, what does your white count do? It goes up. WBC is high. So how do you know to look for these things? Number one, your WBC has to be high. When I'm spitting out WBCs, what's going to happen then? I might have some what? If I'm chucking out lots of WBCs, guys, what am I going to have? Maybe some young ones, some immatures. So immatures, like segs, immature segs, question mark. So what am I saying? I'm going to have increased what? Bands, maybe some metas, maybe milos, I don't know. But definitely my WBC is high, 5.0 to 10.0 times 10 to the third per microliter. So we're talking 15,000, 16,000, 20,000, 25,000, 30,000 WBCs. You probably, if it's an infection, could be cancer. But remember, we're not in malignancy right now. You could look for these things. When you have this, so this is the very big term for today, increased WBCs with, number 2, immature segs or immature forms, we call that a shift to the left. You should be writing that down. That is called a shift to the left. You'll see it every day you work. Every day you work in hematology, you'll see it. Okay? What is a shift to the left? A shift to the left is when you have high WBC count with immaturity. A shift to the left is when you have a high WBC with immaturity. Do you have to have these things? No. Will you? Maybe. Maybe. You might have toxic granulation. That was this. I kind of skipped over toxic gran. Does everybody see how bright those granules are? They aren't faint, are they? That's toxic granulation. So toxic granulation, Dolly bodies and vacuoles. Infection, sign of infection, could be other things, but mainly a very, very strong granule. So again, a shift to the left might have these things, but a shift to the left is defined as a really high white count, and immaturity. Now we also have hypersegmentation. Hypersegmentation is on, toxic gran was on page 136, by the way. And then we also have hypersegmentation. Hypersegmentation is on page I'm looking. You'd think I would know this by heart, but sorry, I just don't retain all this. Anybody?
133.
133. Page 133. If you go there, hypersegmentation, 6 or more lobes. So when you have 6 or more lobes, that's called a hypersegmentation. So does everybody see lots of filaments? This says 5 or greater. The book says 6 or greater. Most of the time it is 5. It could be 6. It depends on your place. Usually, everywhere I've been, it's usually 5. 5 or greater, 6 or greater. 5 or 6, you start looking at these segs and you go, ooh, are these hypersegmented? Now, if you had one, do you do anything about it? No. If you have several, then you path review it, okay? So vitamin B12 or folic acid deficiencies, that's where the nucleus and the cytoplasm are not maturing at the same time. So you get this weirdo maturation because you're lacking B12 or folic acid. You have to have B12 and folic acid in your diet along with iron for your cells to mature, okay? Now, conversely, the flip side is what? What's the issue here? Hyposegmentation. How many lobes do you have there? 2. Does everybody see it only has 1 filament? So, excessive nuclear chromatin, some cells have a round to oval nucleus with dense clumped chromatin. The cell function is normal. It's called Pelger-Hewitt anomaly. What's an anomaly? Have you heard of that word before, anomaly?
An abnormality.
Have you ever been told you were an anomaly? You're an anomaly to me. That's not a good date line. Like, it's not a good pickup line. You're an anomaly. I mean, I guess it could be. I don't know. If you're kind of like Sheldon, maybe, from Big Bang. What's an anomaly?
An abnormality?
It's an abnormality. I gotta get rid of this marker. It's an abnormality. So it's an abnormality. It's kind of weird, but usually the function is normal. Usually. So in hematology, when I say it's an anomaly, it's different, but usually the function is okay. So does everybody see hyposegmentation on page 132? Does everybody see the little different, they look like peanuts, don't they? Don't they look like little peanuts on page 132, it says Pelger-Hewitt anomaly is inherited and affects the majority of granulocytes. Pseudo-Pelger-Hewitt is acquired, and it affects less than 50% of granulocytes and is usually accompanied by other morphologic indications of malignancy, such as myeloproliferative or myelodysplasia. So, what she's saying here is if you have true Pelger-Hewitt, most of the time it's inherited and men have it. It's a Y chromosome thing. So some of you dudes might have it. You just don't know because it's normal. Pseudo-Pelger-Hewitt, what does pseudo mean? Like it. Pseudo-Pelger-Hewitt is not what we're talking about today because we are not talking about malignancy. So in some malignancies you get this, and we'll talk about that next semester. But normally if you start to see this, you're going to start to call them what? What would you call this probably? If you're an untrained eye, you wouldn't necessarily call it a seg. You'd look at the other ones. What do you want to call this? What do you want to call that?
A mono.
You want to call this a band, don't you? You want to call it a band. These are segs, right? So you'd want to call this a band because do you see that it's like a C or an S? Now here you see a filament, but there's only one, so that means it's hyposegmented. So if you start seeing these and you start seeing this, then what do? Run and cry, running out the lab? No, you just look at it and you say, look, this is unusual and you have to refer it to path. The pathologist will say whether there's any kind of malignancy or disorder or not, but more than likely if it was normal, everything else on the CBC was normal and the patient is normal, guess what it's called? Pelger-Hewitt. Because you were born that way. And most dudes have it. Not most dudes. Some dudes have it. It's genetic. It's normal. The function is normal. Now, if you have cancer, you can have that too. That's why you refer it to path. Does that make sense to everybody? So that is May-Hegland anomaly, Shidai-Kagashi syndrome, and Elder-Riley. These are different types of things, and this is in page 206. So if you have Bunny, go to 206. Page 206. Page 206 is May-Hegland. It looks like a Dolly body, but it's not a Dolly body. It's characterized by low platelet count. I'm on page 206, thrombocytopenia, with large platelets and large basophilic inclusions resembling Dolly bodies and granulocytes and monocytes with the absence of toxic granulation. These inclusions are sporadically visible by light, and it is genetic. About 50% of these people don't have any issues at all, May-Hegland, but they do look like Dolly bodies. Going down to Shidai-Kagashi syndrome, these are giant lysosomes. This is genetic. There are very large lysosomes that predominate in here, and so Shidai-Kagashi is on page 207. Does everybody see these big, large gray-blue granules in the cytoplasm? They could be in monos, lymphs, or EOs. Elder-Riley is an azure-philic purple granules. Elder-Riley looks like toxic granulation. Elder-Riley looks like toxic granulation, and that's on page 205. Page 205. Does everybody see Elder-Riley? It looks like toxic granulation, but it is not. They're deep purple to lilac. They're difficult to distinguish from toxic granulation. But if you had toxic granulation, wouldn't you have a high white count probably? This person might not. It's just a precipitation of mucopolysaccharides, and the way it's a disorder, it's a genetic disorder. Okay? But they don't have an infection. They've inherited this. A lot of things are named after the people that they discovered it in or the physician that discovered it. That's how the names come to play. Monocyte and macrophage disorders. This one is beautiful, isn't it? It looks like tissue paper. Doesn't the cytoplasm look like tissue paper to you? This is called Gaucher syndrome. This is an accumulation of glycolipids in the cell. Niemann-Pick is also an accumulation of lipids and sphingomyelin. Sphingomyelin is a type of lipid in your cell. It helps protect your cell. And so again, like glycolipids and things like that. These are on page 210. So please go to page 210. Page 210. I'm sorry.
204.
Two what?
204.
My bad. 204? Okay. 204. Page 204 is your Gaucher cell, is a macrophage, and Niemann-Pick is also a macrophage. Okay? So you're like, are you going to give me these on a smear? I'm not going to give you these on a smear, okay? You'll see these when you get out there in June and July and August, hopefully, all right? I probably won't be showing them here. Could you possibly see toxic granulation Dolly bodies or vacuoles here? Possibly. Sign of infection. High white count, right? Immaturity. Left shift. Shift to the left. Okay, cool? Any questions? You good? Awesome.